So welcome to the session on variant calling. We met yesterday for the QC session, and today we will continue with a variant calling workflow. So you will have been introduced to workflows earlier today and to workflow management systems, I think, as well, such as an NF Core pipeline. But we will start much simpler today, and we will try assembling a workflow on our own using a set of processes to convert input data to intermediate data files and then to an output file. And we'll start by performing variant calling on a single sample, just a very basic workflow. We'll then make that workflow more complicated and extend it to accommodate multiple samples, such as a cohort. And tomorrow, or if you have time to continue with that today, you will further extend the workflow and make it more complicated to adhere to the GATK's best practices for variant calling. And I should say that this is going to be whole genome sequencing based on the same set of samples that we looked at yesterday for QC. Now, our basic, simple, single sample workflow for going from raw sequence reads to a VCF file of variants will be based on running two modules. All right, so starting with the FASTQ files that we looked at yesterday, for a single sample, we will have two FASTQ files representing the first and the second read of every read pair. So these are two matched files representing the paired-end sequencing of a single whole genome sequencing sample. We will perform the first step, which is alignment of the reference genome, resulting in a BAM file. And then we'll perform the second step, the variant calling step, resulting in a variant core format file, VCF file. Now, for this to work out as without more headache than necessary, there are some conventions that I recommend that you follow. First, always create a new output file in each process. Don't overwrite or try to change the input file. Make sure you use informative file names to keep track of what you're doing. And in the file name, include information on the process that you have performed and the sample that is included. And make sure you use the correct file name extensions. That will make a huge difference when you extend the workflow to become much more complicated. You will have a lot of files to work with, and you will not remember what you have done with each individual file. So back to the simple single sample workflow. The first step is going to be alignment to the reference genome using a tool called BWA. Here we will take the FASTQ formatted unmapped reads and map them to the reference genome so that we know where on the actual genome they belong. The reference genome is an assembly of the human genome, a haploid assembly based on a couple of samples from some individuals. And there are multiple versions of the reference genome. Today we will use version 37, also called HG19. And this is a good version that's been around for a long time, since 2009, I think. It's high quality, and many research groups hasn't bothered to swap to a more recent version. But I would say it's more likely than not that you will use a more recent version of the reference genome if you perform these steps on your own data. And make sure you know which reference genome is being used with your data. If you get data that's already been mapped to the reference genome, for example, you will need to make sure that you maintain the same version of the reference genome in subsequent steps of your analysis, or you will end up having a lot of errors. Now for the alignment step, if we let the reference genome be represented by a series of nucleotides like this, we're basically looking to find the position of the reference genome where each read in our data set belongs. This is much more efficient than reassembling the genome individually for every patient. And actually the paired end short read sequencing that we have is not really good enough to achieve a reassembly of the entire genome. So we use the reference genome as a template. In order to quickly and efficiently align all these reads to the reference genome, we use the BWA aligner for Burrows-Wheeler aligner. This aligner depends on a transform of the reference genome to speed up this process. And we're not going to go through that in detail today, but if you're going to spend a lot of time on alignment, then I recommend that you check this aligner, and there are many other aligners depending on what kind of data you'll be working with. When you have aligned your data to the reference genome, your sequence data will now be in a new format called the SAM format. Just like the FASTQ files that we looked at yesterday, the SAM formatted file contains all of your reads. However, it has a very different format. You no longer need to keep two files for a single sample for the first and second read. All the reads will be in the same file. The file has a header section at the top with general information about the sequencing experiment and the aligner and the settings used. And then there's a table section called the alignment section, where every read appears as one line with multiple entries. First entry is the read name, and for paired end experiments, you will see the read name appear twice for paired reads. In addition, as you can see here, we see the reference sequence name. That's the chromosome to which we have aligned this read, start position, length of the read, end position, and the sequence itself, and the Phred score quality that you would recognize from yesterday. So all of this information is collected in the SAM format. Now, we don't actually store the SAM file. For a whole genome sequencing experiment, we would probably have to store several hundred gigabytes of data. So we always compress the SAM file into the binary alignment format, the BAM file. So that's why you will be working with BAM files rather than SAM files, but the SAM format is still inside the BAM file. Now, we'll be working with relatively small files today, but these files are generally huge, and in order to work efficiently with them, we need an index. That's like having a very thick book and you have a lookup table so that you can go immediately to the right page and find what you need, rather than browsing the entire book every time you want to pick out something from it. So the reference genome, for example, which is FASTA formatted, will need an index file. And the BAM file will need an index file to be able to work with them with other tools. The index is a small file that you keep with the actual data file. And there will, of course, be different indices and different procedures for creating the index file for different file types. Even the Burrows-Wheeler aligner requires its own index, which is actually a set of files. Now, once you have created your BAM file of aligned reads, you can carry on with the variant calling step using the tool called haplotype caller. Now, haplotype caller is designed to call small variants, such as substitutions and small insertions and deletions. The type of variation that constitutes a very large fraction of the genetic differences within and between populations, mainly SNPs. Now, having aligned your reads to the reference genome, we often talk about coverage or depth, and that is simply the number of reads that cover a certain position in the reference genome. Now, if we find a mismatch in your sample compared to the reference genome, that may be called as a variant by the variant caller. Here we have an example where we have a C somewhere in the reference genome, but the reads that align to this position have a mismatch where we see an A instead of a C, and sometimes we see the reference allele, the C. So the reference allele, the C, and the alternative allele, which is an A in this case. We have a number of observations of both of them. We have an allele depth for the reference allele and an allele depth, the set of observations for the alternative allele. And if they are good enough for the variant caller, it will make a variant call and put in the output result, which is another text file, the variant call format text file. This file, you will recognize largely the structure of this file from what you saw in the SAM format. There is a header section with general information and a section for individual records, which will now be variant calls instead of individual reads. Here is an example of the content of the VCF file. The header section contains a lot of information on what the individual columns actually contain in the record section. We have the chromosome, position, an identifier if present, the reference allele, the alt allele, the Fred quality score. Just like the quality scores for individual nucleotides in your sequence data, we have a Fred quality score indicating the probability of a variant call being in error. We have an info column, which we will come back to in a little bit. The format column describes the format of the sample column, which is the next column. GT for genotype, AD for allele depth, and DP for depth. So looking at the sample column at the top row, we have a total of 5 as depth, that's the last digit. We have 3 and 2 for allele depth, so 3 of the reference allele and 2 of the alternative allele. That's the number of reads carrying that allele. And the genotype 0/1, that means we have presence of the reference allele, that's 0, presence of the alt allele, that's the 1. So a heterozygous variant call. Now, we're done with the basic workflow for one sample. Interestingly, the VCF file is well suited for multiple samples. We can just add on additional sample columns. The rest of the information can be general for a cohort of samples, including the Fred quality score, which would then be the probability of the variant being an error based on the entire cohort instead of this particular sample. However, analyzing a different sample will give us a different set of putative variants. So we won't have the exact same rows. So we need a way to merge them into a combined VCF. So yeah, we have the variant call file for the single sample, and we are now ready to extend our workflow for multiple samples. To do that, we will perform a very similar workflow individually for each of our samples, and we will be using 3 samples from the 1000 Genome project. First, we align to the reference genome using BWA. Second, we name this. Second, we use haplotype caller with a special option to produce not a VCF file, but a GVCF file, which is a little bit different from the VCF file we just went through. We do that separately for all the 3 samples, and then we collect these VCF files in a combined GVCF step, and then we perform a joint genotyping step to finalize a cohort VCF file. Now, the GVCF file is very similar to the VCF file, but it includes some extra information that will be necessary in order to give a confident result whether there is a variant present or not in samples where you initially did not find a variant. Now that could be due to, for example, a low coverage. So to get the correct result in terms of quality and presence absence of alleles throughout the cohort, and to rescue variants where the number of observations of the alternative allele was very small, we have this VCF file with a lot of additional information stored between the variant site records in the record section of the VCF file. These are not one line per position in the genome. That would be a very huge file. So instead, these are merged into blocks. And we're not going to go deeper into exactly how that works. So this will be part 2 of today's exercise, assembling the variant calling workflow for our small cohort. And the result of that will be a VCF file, just like the first one we looked at, but now containing multiple samples. And for every individual sample here, we see the genotype. 0/0 means reference allele. Then the allele depth, 8 and 0 is 8 for reference and 0 for alternative allele. And the total depth as the last digit in each column, in each entry. Now, today's exercise, we will use 1000 genomes data, just 3 samples, and we have subsetted the data to a small section on chromosome 2. That part of chromosome 2 contains the lactase gene, which codes for the lactase enzyme. And this is an important variant because all over the world, some different variants have emerged that allow us to retain the capability to produce lactase in adulthood. And in some parts of the world, this has become very common due to its positive effects for adults to be able to drink milk. So this is something that is very common in some parts of the world and very uncommon in some parts of the world. And we will take a closer look at one of the most common variants in this part of the world for lactose tolerance. So we will begin the exercise. We'll first make the basic variant calling workflow in part 2, which you will also have time to carry out today. We will extend that for multiple samples. And I would like to remind you to try to keep to these conventions to avoid forgetting what you have done in different files and so on. Part 3, I think, will be tomorrow for most of you, depending on how fast you work. In part 3, we will follow the best practices the current best practices for short variant discovery, which is going to be much more complicated, but also much closer to what you would have to do in a real setting, and what would go into your pipeline if you develop a pipeline using a workflow management tool such as Nextflow or SnakeMake. And we're not going to go through that anymore today. I will have a session on that tomorrow morning. But if you have time, please do go ahead and continue with part 3 when you're done with the first 2 parts.